To the final presentation for the morning session. So after this, we'll have our lunch break. So our next presenter is a graduate of Master of Science in Marine Science from the University of the Philippines, Diliman, and is a candidate for PhD from the same university. He will present his paper entitled Impacts of Aquaculture Nutrient Sources, Ammonium Uptake of Commercially Important euphematoids Depends on Phosphate Levels. Let us all welcome Mr. Bielson Cesar P. Narvardo.
Hello everyone. I am excited to share with you one of our works on the nutrient physiology of commercially important euphematoids. So here, we evaluated the effects of different phosphate levels on the ammonium uptake of three commercially important species, namely Capophycus alvarezii, Capophycus striatus, and Euphema denticulata. So euphematoids may look like ordinary seaweeds, but they are the backbone of carrageenan industry. In the Philippines, euphematoid species include Capophycus alvarezii, Capophycus striatus, Capophycus manesianus, Euphema denticulata, Halimenia, and Betaphycus. And the different types of carrageenan obtained from these different species power a wide range of products in food, pharmaceutical, and cosmetic industries worldwide. Now, beyond their industrial uses, euphematoids are also recognized for their ecological roles. They are known for their capacities to be biofilters in an integrated multi-trophic aquaculture setup or IMTA systems. So here, euphematoids take up excess nutrients from the aquaculture waste stream, resulting to reduced risk of eutrophication. And this capacity to be extractive species is driven by their fundamental needs for inorganic nutrients. So to meet this need, euphematoids, like any other photosynthetic organisms, not only require light, carbon dioxide, and water to photosynthesize, but also require substantial amounts of nitrogen and phosphorus. So these two are considered as macronutrients because of how abundant and essential they are in seaweed's metabolic processes. Iron is also essential as it is involved in the electron transport chain, and some species like diatoms would require silicate in order to produce their shells. Now, how do seaweeds actually take up these important nutrients? So nitrogen is usually taken up in its inorganic form. So we have ammonium nitrate or nitrate. Although some macroalgae can use organic nitrogen such as urea and small amino acids like glycine and alanine. On the other hand, phosphorus is usually acquired as orthophosphate. Now, these nutrients are important because they are used to synthesize essential compounds. For example, nitrogen is assimilated into amino acids, proteins, pigments, and nucleic acids, while phosphorus is assimilated into phospholipids, sugar phosphates, and energy sources such as ATP. Now, in the real world, it's not as simple as just having nutrients around. So the reality of nutrient uptake is a bit more complex. So in the field, the uptake of nutrients by euphematoids can be influenced by several physical, biological, and chemical factors. So physical factors would include desiccation, temperature, water movement, and light. Biological factors would include competition, nutritional history, age, and morphology. Chemical factors would include concentration, salinity, the presence of other nutrients, and chemical species. For this study, we zoom in on one key question: how does phosphate levels affect ammonium uptake in euphematoids, and why is this important? So in mariculture sites, nutrient variability can arise from multiple sources, including the excretion of reared organisms, direct nutrient enrichment from applied feeds, and remineralization of organic matter through microbial degradation. And these processes contribute to fluctuations in phosphate concentrations within the mariculture sites. For instance, in Mindanao, Pangasinan, Philippines, the microbial degradation of uneaten fish feed in an intensive mariculture area has led to elevated phosphate levels, reaching concentrations as high as 4 micromolar during the dry season. Now, the effects of elevated phosphate levels on invertebrates, corals, and phytoplankton have been extensively studied. However, its impacts on nutrient physiology of seaweeds remain poorly understood. In particular, there is limited information on how increased phosphate concentrations influence the uptake of other nutrients. And addressing this knowledge gap is essential as it may have important implications for integration of seaweeds into integrated multitrophic aquaculture systems where nutrient concentrations fluctuate both spatially and temporally. With this in mind, our study aims to investigate the effects of various phosphate levels on the ammonium uptake of three commercially important euphematoids, namely Capophycus alvarazii, Capophycus phreatus, and Euphema denticulata. The approach involves collecting the samples, acclimating them in the lab, and measuring nutrient uptake under various phosphate levels. So first, nutrient-depleted seawater was prepared by stripping off inorganic nutrients from natural seawater. So this was done by incubating natural UV sterilized seawater with Ulva SP inside a temperature-controlled laboratory. The nutrient-depleted natural seawater was then used to prepare seawater media with different concentrations of phosphate and a constant supply of ammonia. The hematite samples were incubated in this media under saturating light levels, and after 1 hour of incubation, water samples were withdrawn to measure the remaining ammonium levels. And ammonium concentration was measured spectrophotometrically. Results of these studies showed species-specific responses pointing to diverse ammonium uptake mechanisms found among euphematoids. Now, several ammonium transporters have been reported in seaweeds. So, for example, the chlorophyte Ulvalinza possesses the AMT1, AMT2, AMT3 subfamilies of ammonium transporters, while the AMT1 subfamily predominates in the rhodophyte Pyropia piezoensis. Now, these diverse ammonium transporters could also occur in euphematoids, which enables them to exhibit distinct responses to phosphate availabilities. Among the three species, Capophycus phreatus exhibited the lowest range of ammonium uptake. And this observation might be a result of the low surface area to volume ratio of the samples used in our study, as shown in this figure. Here, we were unable to directly measure the surface area and volume of the samples. Nevertheless, based on their morphologies alone, we can infer that K. striatus likely had a lower surface area to volume ratio compared to the other two species. Now, our previous study, wherein we were able to have an estimate, these parameters would support this explanation. So in that study, we demonstrated that nitrate uptake was influenced by the morphological characteristics of euphematoids. Specifically, strains with high surface area to volume ratio exhibited enhanced nitrate uptake rates, likely attributable to the greater surface area available for nutrient absorption. In that similar study, our findings also revealed strain-specific differences in Michaelis-Menten genetic parameters, where strains characterized by higher surface tended to display lower Km values, suggesting enhanced nutrient affinity. Higher nitrate uptake rates and initial slopes were also linked to faster growth rates. In Capapicus alvarezii, ammonium uptake was observed even under conditions where seawater phosphate was absent. This suggests that either phosphate is not essential for ammonium uptake or the internal phosphate reserves were not fully depleted during the starvation period and were sufficient to support the uptake of ammonium ions. An increase in ammonium uptake was observed with increasing phosphate concentrations up to 3 micromolar phosphate, and this trend may indicate that phosphate ions are being utilized to generate energy sources that facilitate nitrogen assimilation, thereby enhancing the rate of uptake. However, a further increase in phosphate concentration resulted in a slight reduction in ammonium uptake by seaweed. To the best of our knowledge, this is the first study to report a negative impact of elevated phosphate levels on ammonium uptake in seaweeds. However, a similar phenomenon was observed in terrestrial plants, where high phosphate concentrations inhibit the activity of nitrate reductase through phosphorylation of the serine residues on the enzyme. Now, a comparable mechanism may be operating in these seaweeds. So, at elevated phosphate concentrations, phosphorylation of ammonium transporters in K. alvarezii may have induced conformational changes that impaired their ability to facilitate ammonium uptake. Similar to Capapicus alvarezii, ammonium uptake in Capapicus striatus continued even in the absence of phosphate. However, ammonium uptake in K. striatus increased up to 5 micromolar phosphate. Sorry for that. Although this increase was not statistically significant. Now, this lack of statistical significance might suggest that the relationship between phosphate and ammonium uptake is less pronounced in this species, or that other factors are influencing ammonium uptake at higher phosphate levels. For Ida ditillatum, maximum uptake occurred at 1 micromolar phosphate, but higher phosphate levels resulted to a decline in ammonium uptake. This table provides a summary of the ammonium biofiltration capacities of eukymatoids as reported in previous study. And the data highlight the ability of eukymatoids to efficiently biofilter ammonium across a variety of environmental conditions, including fluctuations in ammonium concentrations and different cultivation systems. These findings underscore the potential versatility of eukymatoids as extractive species in an IMPA setups, and our study contributes to this growing body of knowledge by exploring how variations in phosphate levels influence their capacities as extractive species. So to wrap things up, here's what our study found. So each eukymatoid species responded differently to varying phosphate concentrations in terms of their ammonium uptake. Among the species tested, K. striatus had the lowest range of ammonium uptake across all phosphate levels. Interestingly, even when seawater phosphate levels were depleted, ammonium uptake persisted in the three eukymatoid species. For K. alvarezii and Eden digillatum, ammonium uptake increased with moderate phosphate concentrations, but when phosphate levels were raised further, ammonium uptake decreased in both species. So here are some of my references, and if you'd like to dive deeper into how other ecological factors influence the nutrient physiology of eukymatoids, please check out our list of studies. And before I end, allow me to thank the following: the Algal Ecophysiology team, the UP MSI, the Algal Ecophysiology team, the Mindanao Marine Laboratory, and the OSTP card for my scholarship. Thank you very much for your attention.